Stephen.
Hi, I'm Steve. I'm a neuroscientist, and I study, among other things, Rett Syndrome, which is a disorder of thought or cognition related to autism. And I want to acknowledge the people who did this work with me, my collaborators, Professor Josh Wong, Kirthi Krishnan, and Dr. Billy Lau, who are two postdoctoral fellows in our labs. So I want to explain the title, what I mean by connecting the dots in Rett Syndrome. And what I mean is that cognitive disorders begin with disruptions in genes and ultimately lead to affected behavior. But biologically, there's a long way to go from here to here and a lot of events in between that we don't understand. So if we want to build a pathway from genes to behavior, my lab is trying to incorporate analysis at an intermediate level, which we call the neural circuitry, which is the fine-scale wiring and connectivity of the brain. And we can break down to a simpler problem, somewhat simpler problem, of connecting the effects at the level of genes to effects on circuitry and how those effects on circuitry control behavior. So we can build this path from genes to behavior and hopefully find a point for potential intervention with drugs and other therapeutics. As I said, we're working on Rett Syndrome, which is a neurodevelopmental disorder found in girls. It's caused by mutations of a single gene called MECP2. And one of the characteristics of Rett Syndrome and autism spectrum disorders broadly is a lack of understanding of social communication information. That can include, obviously, language, which is the case for Rett Syndrome patients, and many autistics have difficulty discerning the facial expressions of a girl like you see here. For those of us who are neurotypical, it's pretty easy to see that she's sad in this picture and happy in this picture, but that world of social interaction information is inaccessible, largely inaccessible to many autistics. So we want to study this, the neural circuitry that underlies this symptom of an inability to perceive and appropriately respond to communication signals from other individuals. And in my lab, we turn to our friend, the laboratory mouse, to perform those experiments. And believe it or not, mice are not that different than us in that they have to make social decisions every day about the other mice they meet, and they need to understand who to trust and who to interact with, and learn about the motivations and state of those individuals. And they do that through the perception of signals that other mouse emits, olfactory signals that they smell. And as I'm going to talk about today, these mice also talk to one another with a high-frequency language that we need, is outside of our range of hearing. So one example of how mice talk to one another occurs between mothers and pups in the nest. Can we have the audio? So when a pup gets isolated from the nest, it gets cold and lonely, and it emits this high-frequency, high-pitch whine that sounds kind of like, "Help, Mom." It actually occurs outside of our range of hearing, much higher than we can hear, and I've slowed it down so you can hear this sort of character to it. And this alarm signal is perceived by the female, and she goes and gets the pup and retrieves it. And so we're going to see that in the video now. So I'm going to, in a moment, place a pup in there at a distance, and he will cry out to her, and she goes and gets him. And this is what we refer to as retrieval behavior, and this is dependent on the ability to perceive these acoustic signals that are emitted by the pup and select an appropriate behavioral response. So we developed a behavioral test of the ability of MECP2 mutants. These are mice that are a model of Rett syndrome, that they have their MECP2 gene is altered so that it doesn't function properly. And we compare their ability to learn this task, gathering task from the mother and her pups so that they cohabit with, and compare that to the abilities of the non-mutants, and see if these mutants are impaired in their ability to learn to retrieve pups in our behavioral test. So we're going to show the results of these experiments now. And you can see that after 5 days, a mother is very proficient. So this is the natural mother of the pups. She's very proficient at gathering the pups and is very direct about getting them and bringing them back. And after 5 days of cohabitating with them, what we call the wild type, or the normal mouse, the non-mutant, also does a good job. But the Rett syndrome mouse, the one with the mutated disabled MECP2 gene, doesn't get the behavior at all. So I'm going to summarize a whole lot of work very simplistically and just say that we identified the circuit that's responsible for this, the part of the brain where the wiring matters for this behavior. We localized that to the auditory cortex. And the auditory cortex is the higher brain region that is responsible for processing auditory information, like the calls of the cries of pups. And we found an interesting property to the neural circuit there. We found that there are two neurochemicals that are widely present in the brains of virtually all animals. Glutamate, which is a neurotransmitter that excites other neurons, and GABA, which is a neurotransmitter that inhibits other neurons. And this excitation and inhibition need to be in balance for proper neural function. We found that in the auditory cortex of these MECP2 mutant mice that they were out of balance, such that inhibition was too strong. So we used genetic and pharmacological techniques to rebalance that equation, and this is what we saw with the behavior. So this is a mouse that is an MECP2 mutant but has received our genetic modification. So as you can see, the behavior was completely repaired. So we devised a test for a social communication impairment in mice. We identified the location and nature of the underlying circuit impairment. We identified the auditory cortex as being the important region. And we were able to repair the circuit and restore behavior. And we hope this will provide a molecular and neural circuit target for further study that will eventually lead, hopefully, to treatments and therapeutics for individuals with autism spectrum disorders. Thanks.